Dr. Libin Abraham, after his post-graduation in biotechnology from Ramachandra University, India, worked as a junior research fellows in reputed institutions like National Center for Biological Science and Indian Institute of Science, Bangalore. In 2012, he has been awarded PhD in Virology and Immunology from the University of Heidelberg, Germany. After his PhD, he worked as postdoctoral fellow in the University of British Columbia, Canada. So his research interests are virus pathogenesis, lymphocyte signaling, and activations. So in this current scenario, it is very important that this kind of talk will be more helpful and create great awareness. So I would like to welcome Dr. Libin Abraham to preside over the presentation. Dr. Libin.
Hello, everybody. Good morning. Can you hear me?
Yeah.
Okay, I'm going to hear me properly. I'll get started. So I want to first thank the organizers for giving me an opportunity to share some of the thoughts about the coronavirus. And based on the evidence we have gathered so far, how exactly we are going to tackle the virus and how our future is going to be. So in order to discuss a bit about the future of the virus, it is very important that we have to know the history of the virus. And we have to know what we are going through. So it will be kind of awkward that if I say, okay, this is going to be a future, because we need to understand what happened in the past and how we are going through the current scenario now. So back in 2017, there was this cover article in Times Magazine. And as you can see from the cover, it says that, warning, we are not ready for the next pandemic. So this was based on one of the very famous TED Talks Bill Gates gave, where he told that he's not worried about nuclear weapons destroying the world or any new war. What he's really worried is about a respiratory virus, which can be very, very bad. So similar opinion was also shared by Dr. Anthony Fauci. Some of you might already know Anthony Fauci by now. He's the director of the National Institute of Allergy and Infectious Diseases at NIH in USA, and he's also in the task force for the President of the United States to advise him on how to handle the coronavirus. So he's a very experienced scientist, doctor, and also a public health spokesperson. So when he was asked last year in May about what does he worries the most, so this is what he told. So I'm going to read what he exactly said. I am most concerned about the emergence of a new virus that the body doesn't have any background experience with, that is highly transmissible from person to person and has a high degree of morbidity and mortality. So he was actually not telling about influenza. In fact, he said that it could be something like SARS. Just fast forward to today, this is where we are. So we had already enough evidence from the past that we were already progressing towards a pandemic. We largely ignored all these warnings. So this is not something new for many of the people who are already in science. But now, since we are all of us are now locked down, we feel very bad. So what exactly is a coronavirus and COVID-19? So already Abhilash has clarified this, but in case any of you is not very clear with what exactly is the name of the virus, it's a SARS-CoV-2. And this is the name given by the ICTV, which is International Committee on Taxonomy of Viruses, who is the one who is a official authority to name viruses, which is based on the genetic structure of the virus. And obviously the name is given by WHO, and both of them happen on the same day, on Feb 11th. The name of the virus was given, and also the name of the disease was also given. So why did we rush to give this name? Because we found that a lot of the symptoms we had for the new virus resembled our previous SARS virus symptoms, which we had in 2003, 2004, which was a SARS virus, which is also a coronavirus. And there was a time where everybody was calling this as a Chinese virus or the Wuhan virus, which was kind of a bit racist. So it was very important that the ICTV has to come up with a name so that everybody can call this. So just to briefly tell a background about coronaviruses, like these are positive sense single-stranded RNA viruses, and these are enveloped viruses. So for those of you who already have learned about viruses, you know that there are two kinds of viruses: the DNA viruses and the RNA viruses. And you also have the enveloped and the non-enveloped. And if you think about the genetic material of the virus, it can be either a plus sense or the minus sense, depending upon how you wanted to code the information. So we first discovered coronavirus in farm chickens back in 1930s, but the very first human coronavirus was discovered in the 1960s. It was actually from a common cold. And we actually call this virus 229E and OC43. These called very mild cold symptoms. And the famous Scottish virologist, her name is June Almeida, who actually did the first cryo-EM of the virus, gave us an understanding of how this virus is. So shown on the left side of my screen is basically the graphical representation of the coronavirus. And you can see that this very spiky feature, which is why it's called the corona, or the spike or the crown. And you can see that this is the original image from the 1967 paper, which from here we rendered to this image. She is a very experienced cryo-EM person, and it was a leading person in the field at that time, doing a lot of cryo-EM, a lot of EM for virus studies. So then we had a lot of new viruses coming. So in 2003, we had the SARS-CoV. In 2004, we have NL63. In 2005, we have HKU1. And much more recently, we had in 2012, the MERS virus. And then we have the SARS. So everything I showed in yellow in the screen are actually very, very mild. So this causes very common cold or something like that, and this goes away. And everything in the red, which is basically the SARS virus, MERS virus, and the SARS-CoV-2, what we have now, the COVID, they are pretty nasty. So if you look on the structure of the SARS-CoV, as I told you, it's a coronavirus. The most important thing about this virus is a spike protein. So it's very tough to navigate the coronavirus without explaining enough the spike protein, because we are trying to develop vaccine using the spike protein. We are trying to target the virus entry using the spike protein. And this is a very essential part of the virus by which the virus enters the cell and also mediates the transmission. So shown here is a schematic representation. Again, you can see that the RNA is coded inside the shell. And if you take the spike protein, it has basically two subunits, S1 and S2. And it has a region called RDB, which is called the receptor binding motif or receptor binding domain. So which basically the coronavirus spike protein, RBD, binds to one of the host cellular receptor called the ACE2, and most of you might know already by this now. It's called the angiotensin converting enzyme 2. What is more interesting is like how this spike protein exists. So here is where all the confusion comes. So I'm showing you is the most recent cryo-EM structure of the coronavirus spike protein. It's a trimer of 3 spike proteins. So basically, when you think about proteins and when you think about the basic level of proteins, start from amino acids, and then you have the secondary structure, which is basically beta sheet and alpha helices. And then you have a tertiary structure, which is a 3-dimensional structure, and then you have a quaternary structure. So what you have here is something more like a quaternary structure. You have 3 spike proteins together, which is extends a trimer. The interesting part about this is like all of these protomers, the individual monomers of the spike protein, have the same amino acid sequence. However, they have a different conformation. So this is very interesting because shown in the left here is basically a closed conformation, and shown on the right is an open conformation. You can see one of the RBDs is opened. And if you try to overlay both of them, you can see in the graphics that like there's a closed conformation and that's open conformation. So what is unique about this spike protein, the coronavirus, the novel coronavirus? So basically it is binding to the same receptor ACE2. It was the same receptor used by also the previous SARS virus, but here there is something interesting. So we have a cellular protease called furin, which basically cleaves these spike proteins and thereby exposes the remaining 2 residues. So as a consequence of this, most of the studies have shown that the new coronavirus spike protein binds to the receptor with much higher affinity than the previous 2003 version. So something which Abhilash was telling, like, all the confusion happening, what is the difference in structure? Why is it binding differently? This is a reason. So the receptor binding domain has acquired some specific mutations. And it is also taking the help from a cellular protease called furin. So it has a furin protease cleavage site, which allows a virus to open up their spike protein and have a better attachment to the receptor. So if you go to the genome, it's pretty simple. And again, this might be beyond the scope of today's audience. Some of you might not be interested, but the long story short is, this is a very large genome. So if you think about RNA viruses, the most common things that come to your mind is basically influenza viruses or the HIV. So HIV is entire the length of the entire HIV genome is close around 10 kb. So it's only 1/3 the size of the coronavirus. And influenza is, again, only half of the size of the coronavirus. So this coronavirus is actually packaged a very large genetic material, close to 30 kb. And it also codes for a lot of proteins, including non-structural proteins, which is shown here, NSPs and NSPIs. And it also has an RNA-dependent RNA polymerase in order to just allow the copy the viruses. And you have the famous spike protein here, which is the structural proteins, and you have accessory proteins as well. So these are large viruses with a very large genetic material. So ignore this slide if you're not interested, but this is purely for the interest of those people who want to know more about how the virus enters the cells and how the virus exits the cells. So as I told you, the virus binds to the ACE2 receptor. It internalizes after receptor-mediated internalization. Then it uses our host machinery, which is basically the ribosomes. So the translation is happening, and it's a positive sense RNA. So immediately you make the RNA-dependent RNA polymerase. Now the virus makes copies of itself. It also basically makes virus proteins. Those virus proteins which go to the envelope is basically going to secretory pathway through the Golgi, to the ER and Golgi to the cell surface. But those made inside the cytoplasm, like the N nucleocapsids, are assembled here. So you have viral assembly happening, and the mature virus-like particles or virions are budding off from the cells. So keep in mind that the envelope of the virus is basically from our host cell membrane, pretty much. So this is about the life cycle. And now comes the more interesting questions, like, we are all very frustrated about the new virus because, as I told you, like in the scientific community, we know this was happening, but now people didn't expect like something much severe like this. So where is the real origin from? So shown in the schematic is basically like a graphical representation which was made probably in the very beginning of the SARS-CoV-2 virus, and hence there are some problems in the schematic which I'll explain. So everything we believe is originated from the bat coronaviruses. So if you think about the 2003 SARS virus, it had an intermediate host, which is the civet cat, and then we got it in the humans. If you think about the 2012 MERS, it was the camel, the intermediate host. But we cannot say yet with the confidence what is the intermediate host in the new coronavirus. So the very early studies suggested that it could be an intermediate host called the pangolins. So pangolins were actually trafficked into the Wuhan wild market, and there was a good chance that the virus would have been got from the bat to the pangolin. And some people even speculated another intermediate host like a snake, and then family to humans. So we really don't know. That's why all these question marks are here. We still really don't know what are the real intermediate hosts. But at the moment, let's leave it for the time being there. So if you ever think that it's not just the coronaviruses or the SARS which the bats carry, right? So the Kerala has already faced Nipah 2-3 years ago. So we have the notorious Hendra virus, Ebola virus, Marburg virus, SARS virus. So the bats are like the super hosts. So it's always good to think like, why? Why are bats able to carry so much viruses and nothing happens to bats? So the obvious question in the scientific community is like, why does bats live with so much viruses? And why is it such a super carrier? And we can also ask the question another way. Why are these viruses so harmful for us? Like, we sometimes get the bird flu, or sometimes we get from other, like from the swine flu or things like that. But the virus which we get from the bats are much more severe for us also. So this is very important to understand like what exactly is happening. So there is a simple illustration to explain this, but the best thing to keep in mind is that this is basically due to a co-evolution of the bat's ability to fly. So, bat is only flying mammal. So the bat's ability to fly and the ability of the immune system to kind of ignore some of the host inflammatory responses. So it kind of finds interesting. I will go through the schematics. So the only flying mammal is bat. And you can see that it's a very high metabolism. And due to very high metabolism, you create a lot of free radicals. And the free radicals actually cause cellular damage. So this is very important because the minute you have the cellular damage and you have free segments of DNA inside the cell, if you have a fragment of DNA floating in the cell, the body will respond. There are many sensing mechanisms called DNA sensing mechanisms in the body, which will suddenly recognize that, oh, why is there a piece of DNA in our cytoplasm? So we need to respond to that. So basically, this is actually caused by a pathway called the gas sting pathway. So you can see some fragments of DNA here, and you see the gas protein, and you see subsequently the sting protein. It actually causes interferon beta production and so forth. But however, in the bats, the sting proteins are actually mutated, thereby dampening the functionality. So yes, it do produce host inflammatory responses, but the response are much more reduced. So it's not aggravated. And hence it is a reduced inflammation. The virus can still survive in the bats. Okay. At the very same time, the bats have evolved with very unique antiviral immune responses. So they have specific cells which can actually attack the viruses. They have the different gene expression which actually also can control the viruses, which is completely absent in most of the other mammals. So not only is that like it is like a balance. Like you want to attack. Bats have very powerful immune system and it attacks the virus. At the very same time, it doesn't elicit exaggerated host inflammatory responses. And this is very important to keep in mind because most of the people who actually have severe COVID or ARDS, respiratory distress, is mainly because of the host inflammatory responses. So we actually overreact to that. So the bats actually have neutralized that part. And it is also believed that like the bats, actually they form actually the 1/4 of the entire mammalian species. There are so many different species of bats and they all are present in almost every continent except Antarctica. So the coexistence of all these bats allow the viruses to recombine and mutate, and they create new strains. So we have like hundreds of coronavirus in bats. And as I told you earlier, the schematic is good to understand. These viruses which are kind of happy and killing the host, most of the like humans or anything which is walking on the ground, but bats actually fly. So it actually increases the basal metabolic rate, or the temperature increases and the viruses die off. So just think about some viruses which can actually tolerate these high temperatures, like a virus in the bat which can survive these high temperatures. And if that virus spills over to humans, there's where the problem starts. So that's why we actually have a severe fever, and our body is trying to raise the temperature to kill the virus. But these viruses have already been evolved in a much more powerful species, and they have already taken care of that temperature part. So it's good to think about how the bats are very unique in terms of harboring so many viruses, the unique immune system they have, and how viruses also co-evolved according to that. And hence, anything we get from a bat can be very, very bad. It was not the case all the time. So bats are really not responsible for all these issues. If you think about that, we humans and bats coexisted for a very long time. And it was all fine. It's until early 1960s, we started seeing that we are slowly getting human transmission. And shown here is a little schematic to show that, over the time, we have more and more sick people, and we're getting more and more, zoonotic transfer, from bat to the humans. Why? It's a very simple answer. We have been now going to the bat ecosystem. So the bats have been living in an ecosystem, and we have been trying to have farming there. A good example is in Malaysia, where they have this swine farming or the pig farming, and a lot of this, the natural bat habitat has been destroyed, and hence the coronavirus has got spilled over from the bats to the pigs. Similar thing happened also in Australia, where they removed a lot of bat ecosystem to build houses, and bats used to come and eat the fruits from our trees and so forth. So this point. So if you think about what happened in Kerala, since we still don't know what is the ground truth, but it could be possible that a bat which has been in its natural environment has been kicked out, and probably they start coming to our habitat, and then it would have been like cross-contaminating the fruits which we have been eating and something like that. We don't know yet. Probably it's a case. Okay, that's pretty much about that. But some people are not happy with that explanation, because everything I told was about bats, and then you're like kind of convinced. But if you think more about the true origin of the SARS-CoV-2, there's a little of speculation. So I don't want to take too much of your time here, because there is not too much data here. Everybody believes it's a natural evolution which happened in the bats, as I just explained. The viruses are constantly challenged and they evolve over time. So there has been a very good example to explain this. You have either natural selection in an animal host before it went to humans, or you have a natural selection in the humans then following the zoonotic transfer. The paper which discussed all this came in March 2020. It was from the lab. The lead author of the opinion piece is not actually a paper; it's an opinion piece. Is by Robert Gary. So Gary was in a pressure to kind of get the word out that this is not a laboratory created virus. So hence he wrote this, and you can see that how many people already access this. Like 4.64 million people access this. So many citations, and this has been well downloaded paper. So people use this opinion piece as an argument to say that, okay, everything happened in bats, we're all good. But there's a catch. If you look here, the same spike protein and the receptor binding domain of the spike protein, there is this polybasic cleavage site, which is basically the furin proteases use. So you can see that if you look on the SARS-CoV-2, you can see these amino acids here, new amino acids. You don't see this in a pangolin, which is supposed to be the intermediate host. You don't see this in the bats or the first SARS coronavirus or any other bat-related coronaviruses. So this is something very novel. So as I told in the beginning, the spike protein has this novel furin cleavage sites, which allows them to open a little more better and binds to receptor much more efficiently. So the transmissivity is much more aggressive than the previous version. So in the opinion piece, what Gary wrote was like, hey, all the evidence shows that the new virus is much more stronger. So it will speculate that probably, this could have been evolved maybe in a lab. But he says that the modeling suggests that it is very tough to have these mutations, or it's very tough to have a confirmation, which is very tough because you do real experiments and you show that there is high binding affinity. And then you go back to modeling and say that, hey, maybe modeling says that it's not easily feasible. That's not a clear explanation for scientists. So many people really criticize this opinion piece. And what is my opinion on this? Like, I still don't know. Like, this is a very valid point. We still don't know how it happened. Interestingly enough, in the same paper, Gary wrote that these mutations can be acquired just by growing in the lab for several passages. So if some of you are familiar with growing cells in cell culture, you know that like you grow cells over time, you change the medium and you grow it over days and months over time. And every time you do that, there's a small changes in the cell lines. It happens. Same thing happens with viruses. When you keep passaging the viruses over time, you basically have changes. So it's already shown in influenza virus that if you keep growing in culture for some time, they immediately acquire the very same polybasic cleavage site. So it has been shown in a laboratory setting that this can happen. So they are kind of elusive. And in this opinion piece, they didn't really discuss about that in great detail. So all he said was like, well, we find all this, but modeling suggests it may not be the case. And that's a problem because modeling is not always true. Anyway, there's other crazy theories about this. Like, we have a crazy scientist, who actually artificially created in the lab for the purpose of bioterrorism. I mean, I don't really buy that because there's not much evidence on this. But there is something in between called still a natural evolution in a lab. So like you are in a truth triangle now. Like, you have this crazy idea of bioterrorism. You have the natural evolution in bats or natural evolution in humans, but everything happened naturally. Versus a third scenario where it could be naturally evolved in the lab. And one cannot completely ignore this possibility. And you are somewhere in between, thoroughly confused. So the reason why we think that it could still be possible from a lab, I'm not suggesting it is, but it could, based on a preprint now available in bioRxiv. So this group of scientists from the Broad Institute at Boston has taken the SARS-CoV virus and also the novel coronavirus. And what they're showing here is a nucleotide substitution, so basically mutations. And you can see is that you can see two waves of this. Was it January to March in 2003? And there is a second one from February to May. As you can see that there are so many substitutions happening over this, that's like almost 70 substitutions. But if you look on the coronavirus which we have now, they are not doing much in the last 3 or 4 months, which is a good news. But it seems like it has been already been adapted in the lab. So the speculation in the paper is that it is possible that a non-genetically engineered precursor could have been adapted to humans while studied in the laboratory. And such a possibility cannot be ignored, it's having a precursor. Another interesting thing, and again, I'm not going to spill all the controversy theories, but something to keep in mind is like, in Wuhan, you have the biosafety level 4 laboratory in the Wuhan Institute of Virology. That every country gets a top national biosafety level 4 laboratory to do extremely pathogenic work, right? So it was in Wuhan, very close to, again, the pet market. And the Professor Xi Zhengli she's called the bat lady because her whole research was to understand bat coronaviruses and what is the chance of these bat coronaviruses spilling to humans. So she has been doing a lot of recombination studies, and she even showed that, it published in a Nature Medicine paper, that a modified virus can replicate efficiently in a primary human airways and achieve in vitro titers equivalent to an epidemic strains of SARS-CoV-1. So she has generated this. This information was there in the Wuhan Institute of Virology website until April early, and then they removed it because of some reasons, because people started thinking all the conspiracy theories that they did on purpose. I'm not telling they did on purpose. Like, scientists do recombination and gain of function experiments on every model all the time. That's how you study something. You have either you introduce a new gene function and see what happens, or you remove the gene function. So the gain of function is basically the gain of a function by putting a new gene, or loss of function is by gene silencing experiments, or you do recombinations. But it's a little more challenging when you have something very pathogenic like bad coronaviruses. So basically there is a bit of confusion there. And again, I'm not suggesting that it is being created for purpose of bioterrorism or anything. There's enough controversies out there, but you cannot completely ignore a possibility that there's a spillover. Did it happen before? Yes. So back in 2011, there has been an enhanced strain of the bird flu virus, which has been developed, and it actually created a widespread alarm, and people have to immediately stop the research and completely clean out the space. So we have created much more pathogenic virus than it naturally is. But the good management of it, proper waste disposal and everything was able to completely clear out that. So such things all happen. So where am I personally standing in this truth triangle? Where I'm somewhere here. I still believe it's a natural evolution, but you cannot rule out a possibility that the same natural evolution could have happened by growing all these viruses in the lab. Especially because we are trying to modify the coronavirus to infect mouse, to infect other species. And it's always, the minute you modify it, the chance of spillover is possible, and then if you don't properly discriminate, it goes over. Okay, that's enough of the conspiracy theories for me. You can go straight to the transmission. So you have heard a lot about how this coronavirus transmit. So the long story short is, it is basically transmitted by droplets or by aerosols. So that we all keep social distancing, okay? And that is, the suggestion is from the CDC is like 3 to 6 feet or 2 meters, however it is. And this has been a new norm. But there is a problem in that. The biggest problem in that is like, whenever you talk or if you sneeze, you basically not only create droplets, which is shown here in blue, which fall down, which are the big droplets. You also create something called, the gaseous vapors called the aerosols, which are much more smaller than the droplets. And the problem with these aerosols is that it can still be present in the air for like at least up to 3 to 4 hours. That's the maximum duration of the time they measured. And this can be potentially bad. So just to give you an idea, droplets are much more smaller than the size of your hair. So you're not going to see a droplet. So you're never going to see an aerosol. So the aerosolic transmission has been completely undermined previously. So even though we keep social distancing, sometimes you can still transmit just because you're talking or you're sneezing, and aerosolic transmission. Or think about that. So you are an infected person. You go into an elevator or into a lift, and you just sneezed in the lift, and then you came out, and somebody else go to the same elevator like after 15 minutes, and depending upon how long the aerosolic particles remain, there is still a chance that you can have it. So you didn't even see the person who entered the lift. You didn't have any direct contact, but it's still possible. So this is something which has to be keep in mind, that you have an aerosolic transmission, and you can clearly see that the gas is emitted when the person coughs, sneezes, or even exhales. This is bad. So the CDC have already suggested that you need to wear a mask in the public, and that's what most of us are doing, which is a great thing. You know the two types of masks, surgical masks, which most of us are using, but keep in mind that that is not going to protect you, because that is, your droplets can easily enter into these voids in surgical masks. If you want to really protect yourself from getting coronavirus, you need an N95 respirator, which is not easily available, mainly because it is used by the healthcare professionals in the ICUs and in emergency wards, because we are directly dealing with corona patients. They are doing the ventilation, they are putting the ventilatory equipment into their trachea. So all these things create aerosols, and they need N95s. We cannot use it. That said, it is still good to have a surgical mask, because in case if you are infected, you can prevent it. You put a mask there, the aerosols get to the sides or so it doesn't go to the larger distance. So mask is very good. And I will come later in my talk that all the countries, in fact, who was using the mask, mainly the South Korea, Taiwan, they were able to really manage the epidemic. And this is one of the success stories of managing that. So that one thing is about all the aerosol transmission. And now you know about cleaning the hands. So you think, why cleaning hands if everything is aerosolic? The main reason is because after you sneeze or after you cough, these droplets will fall down and in different objects. You can see here, shown here is a amount of virus viable in copper, cardboard, stainless steel, plastic. You can see that they all exist for different time. So basically, glass is the worst. Like if you have a glass of water or something, or if you have plastic, it can be there for a longer time. So all these are approximations. Keep in mind that these are experiments done in a controlled setting without airflow. So if you're in a different environment, it's different. So in this slide, I want to bring in attention for something else. You can see that the droplets actually fall down. They are bigger in size. And you can see the airborne transition by the aerosols. You see that smaller droplets evaporate and virus continues on. So something to keep in mind is that, like, the more the temperature is, the more hotter the region is, there is a slight chance that you can reduce the aerosolic transmission. It is not shown for the coronavirus yet. At least it has been shown for the seasonal flu virus, the influenza virus. So the influenza comes in waves. Whenever there's a winter time, at least in North America, in winter time, or in Europe, you have the coronavirus, you have the influenza virus coming. And by the time you are going back to the, just a minute. Sorry. By the time you go back to the summer, again, it goes up. So people think that the temperature, basically the reduced transmission by aerosolic is the main reason. Again, we still run the evidence that the coronavirus is seasonal. We have no evidence yet. But one of the probabilities why we have less infections in one of the warmer countries could be that. We don't know. About infection, it comes through the majority of the droplets come through the nasal route. You can also have it scratching your eyes, or you're touching your mouth. So mainly the oral routes and the eyes. Once it enters, you have a phase of asymptomatic transmission. So this is something very unique about this virus. So some of you would have heard about a Shailaja teacher talking about, Health Minister of Kerala talking about how we handle Nipah virus. One of the key things about Nipah virus is that Nipah virus, the virus shedding from the person to person happens really in the late stages of the virus, when the person is extremely sick, having this, hemorrhagic fever and everything. It is during that peak phase it is really transmissible. The problem with the coronavirus, the new coronavirus, is that it is like a silent transmitter. You have an asymptomatic phase. I can be sick. No, I can be having the virus, but I'm not sick, but I can still transmit everybody. This is a big challenge in modeling and making all the predictions. So we have not seen something quite like this yet. So we see here, you have the number of days, the 14-day incubation period. Some people are completely asymptomatic for 14 days, and they are still shedding the virus. So these all things are all kind of approximations. These are not don't take it as a standard that everybody has symptoms at 5 days. Not like that. It changes. But you can see that the first few days are asymptomatic. Then you have symptoms appear, depending on mild symptoms, and then you have a quarantine period. So the first, most of the symptoms you get is basically, you have the fever, high fever, you have the headache, you have the muscle pain, you have shortness of breath. And as again Abhilash pointed out, like majority of the people, it just goes like a regular, like a flu. It just goes away. Like here it is shown 80% of the people with mild cases. I would still call it asymptomatic / mild. It's completely is okay. Some people get it a little more severe, and some people get critically sick. So we have seen that the most people who actually fall critically sick are having one or two comorbidities, like either diabetes or cardiovascular diseases, or maybe asthma or things like that. And also, obviously, these diseases are also associated with age, and hence many people who are older, again, their immune system is weaker, and they also have one or two comorbidities, and hence they fall sick. That doesn't necessarily mean that every old person is at a high risk. No, there are many old people who actually recovered completely. They're all fine. Also, young people are not completely immune either, even though they have a very healthy immune system. There are people who fall sick on a Monday and they died on a Wednesday. That bad. So, you cannot take anything for granted because we still don't fully understand the virus. But we have to keep in mind that, like, for some people, this is like a normal thing. And for some people, it's very severe. And in order to fully understand this, you have to think this from the perspective of how much virus has been entered the body and what is a natural immune response of that particular individual for that virus. So this explained a bit of explanation in the immunity or immunology. I'll come to that briefly later. But keep in mind that most of the people are symptomatic or have mild symptoms. In some severe cases, obviously, they say the viral pneumonia. The problem is that it just don't affect only the lungs. And again, Abhilash told this in the introduction. So we just think that, like, okay, it's a pneumonia and then we have a difficulty in breathing, that's it. No, that's what we thought in the beginning. In January, we thought that's the case. But we have found that it actually affects a lot of organs. So basically in the lungs, you have basically, the entire inflammatory responses. You can see all the immune cells are there. The alveolar sacs and the cells are actually broken because virus actually keeps shedding from there. You don't have the proper oxygen transfer in the alveolar space. That's how the lungs are being affected. But you also keep in mind that the virus actually binds to a receptor called the ACE2 receptor. And these receptors are mainly present in the lung epithelium and also in the intestines, but many other cells also has it. In fact, we have found that it can actually these blood vessels, and then you can see that it actually forms some clots. So most of the people who have severe clot, and people don't fully understand why the dysregulation of the endothelial cells after virus regulation and clot-like structures. And this is also the reason some people have COVID toes. The toes become swollen, and that's because of they think that they again validated. They think blood clots in the toes of the patients. You also have problems because it crosses the brain barrier, the brain. Some people have seizures, confusion, brain inflammation, and you can see people some of these cases, and you have the nose. Have you read that, like, the people who had COVID or who had coronavirus, they suddenly lost the sense of smell? And this is kind of easy homemade diagnostic test to see that. I mean, you can still lose your sense of smell for many reasons, but people were telling that if you suddenly lose your sense of smell, then probably you might have coronavirus. The reason is that somehow many people lost their sense of smell, and they think that is because of either the nerve endings or because of other things. Again, the liver damage, kidneys, and intestines. As I told you, intestines also has a lot of ACE2 receptors, and then it actually causes diarrhea in many people and gastrointestinal problems and so forth. So it is actually like, it's not just the lungs alone or an upper respiratory tract. Even though it's a respiratory virus, the damage is pretty extended and broad. So we have certain stages of infection. And this is something which I told also in the beginning about host inflammation, giving the example of the bat immune system. So we have the stage 1, stage 2, and the stage 3. Stage 1 is an early infection, stage 2 is a pulmonary phase, and the stage 3 is a hyperinflammatory stage. So you can see that this is a viral dose, and you can see that as you have the virus response phase actually slows, the amount of virus actually slows down, okay? And you can see the severity of the illness is also climbing up. And there is a phase where our body overreacts, which is basically the hyperinflammatory phase. And this is a time where exactly things go very badly. As I told you, like there's a damage from both sides, the virus infecting ourselves and the way our body react to the virus. So it's like a mixed thing. And as I told in the previous example from the bat immune system, the bat's immune system doesn't overreact. At the very same time, they have a very powerful immune system as well. On the contrary, ours is very different. Some people give very severe post-inflammatory responses, and that itself is sufficient to damage the system. Yeah, they does not mount post-inflammatory responses. So why some people are getting sick, why some people are not getting sick, all depends upon every individual's ability to respond to something foreign. And it's something which we cannot put like a broad category. Okay, you are going to get sick because of immune system. It's tough to say, because the way our immune system responds also depends upon a variety of factors, like right from our genetics to the food we eat, to the bacteria in our intestines, to a lot of other things. So you cannot just categorize everything. But keep in mind that by our own process. So how can you tell there's a SARS-CoV-2? Well, you are getting some symptoms, and some of the symptoms which you have is a high fever, you have the head and throat pain, you have the shaking chills, runny nose, diarrhea because it affects the intestines, you have confusion, cough. These are all the common symptoms you have, which can suggest that you can have coronavirus. But at the very same time, it's good to think whether did you travel recently, or did you wash your hands properly, did you wash your vegetables properly, or did you have your mask contact somebody who was probably infected coronavirus. So like you don't get the doctor, you ask your symptoms, and then they will give you some advice. And after hearing over phone, we have to go to the special coronavirus or COVID hospitals or clinics to get evaluated. So many people just come back, tell me, oh, it's just a common flu. Okay, they don't have any. So the only way to say this with absolute certainty is by diagnostic test. And we have many tests. I just want to briefly test this. We have the molecular test, which is called the PCR test. We have the antibody test, and we have the antigen test. So basically, it's very simple. The PCR test, the polymerase chain reaction, you detect the virus itself. So it's very simple. You take the RNA of the virus, you do the polymerase chain reaction, and then you find it. Then you take a nasal swab. Some people are now using the saliva swab because I don't know again how this is done in Kerala and India, because here the technician or the doctor is not taking your sample. They give you the swab, and the patient is instructed, or the suspected patient is instructed to how to do a nasal swab properly, and then spit out, and then you give it back to the doctor. So the procedure is done completely by the patient itself. So what if I didn't take a proper swab? I can have a wrong result. So it's from nasal swab, and then you detect for the viral RNA material. The antibody test, on the contrary, as you most know, the body will produce antibodies, and you do the antibodies. And it's a blood test, and it doesn't confirm the presence of virus. All you can say is whether you are infected in the past, and depending upon the nature of the immunoglobulin or the antibody, whether it's an IgM or IgG, or different types of that, you can say it's an early or late and so forth. But that is not yet the conclusive test. The conclusive test is always directly detecting the virus itself. The third test is the antigen test, which is a little more popular, at least here. I don't know how it's back in India. Here, again, you're taking the virus again, but instead of the RNA, you're taking the virus protein, like mainly a spike protein. And then you're doing an ELISA kind of assay, like basically you're detecting by an antibody-based method. This is not very sensitive. It can also have some false positives and everything, but you get a result pretty fast. So that is something which people have been using to do some quick testing, so forth. So the only confirmed test is a PCR test. And this is the word social distancing we have been telling. And from the aerosolic spread, you might probably know that, probably social distancing alone is not sufficient. You need to probably also, wear the mask and clean the hands and so forth. But for people who are not fully understanding social distancing, this is a good way to explain that. The flattening the curve, and I'm pretty sure that most of you know this. So the whole concept is that, you have this the spike of the cases happening. You have so many people getting infected, but you have a limit in which your healthcare system can actually accommodate. And when you have too many people coming in, then you just have to let the people die. So in the past, when somebody dies, it's because, okay, think about somebody who had cancer. So they have a terminal cancer. We tried chemotherapy, we tried all the drugs, and nothing worked. So we will say that, yeah, it's his time, it's her time. So there's nothing working. We used all our resources, all our medicines, all our infrastructure, and nothing could save the person. And we let them die. But when the coronavirus came, it was not the case. We still could save a person, but the person was not able to access the necessary healthcare, access a ventilator when needed. So people were dying because of the demand. And that is the reason we need to flatten the curve, where you stay home and you try to give the social distancing and do all this practice. So you still have the virus in the population, but you just kind of like flatten it all the time. So a bit of immunology here. So I've been asked how to explain a bit why some people have these different responses. And as I told you, like, it's very complex to explain, but if you think about how our body is fighting any virus or any foreign organism, you have to think about what our body is exactly doing with the immune cells, which you can broadly classify as innate immunity and adaptive immunity. And most of you know this from your, from your high school, or maybe from your pre-degree studies itself. So we have the innate immunity, which is much more rapid. We have a lot of cells for that, dendritic cells, macrophages, and all the antigen-presenting cells. And these immediately clear the viruses in a much more broader manner, rather than more specific manner. But it doesn't have any memory. Okay, innate immunity doesn't have any memory. Or the adaptive immunity, on the contrary, mainly consists of two major cells, like the B cells and the T cells. And the B cells make antibodies, and the T cells, the two types, CD4 cells, which is the master orchestrator of all the regulations happening in the immune system. It's a CD4 cells the HIV infects, if some of you might know, and that is how the HIV completely messes up our immune system. So the CD4 cells, which is like regulating everything happening in the body, and the CD8 positive cells, which are basically cytotoxic T cells, which kill the virally infected cell. So keep in mind that if there's a cell which is infected by a virus, or if there's a cell which is actually become tumorogenic, or like become a cancer cell, there's only one fate for the cell, basically cell death. And the death is mediated by either a killer cell, natural killer cell, or a CD8 positive T cell. So basically you need to keep killing all the infected cells. It's not like a repair mechanism, like, a virus inside the cell, you're going to repair it over. That's not the case. So everybody's body is different. Everybody's adaptive system is different. Theoretically, we could make an adaptive immune response to any three-dimensional foreign object, any three-dimensional protein. So we have like a very vast repertoire in our body. Like you can make like for anything, literally. But it all depends upon how much is being administered and how exactly you are kicking in your response. So that is why vaccination sometimes, even though it gives a long memory for some people, some people it doesn't give that long memory, because everybody's body is slightly different. So, but even though you say this theoretically, sometimes it doesn't work like that. So maybe I might have an immune response based on some previous antigens I have seen, like a foreign object I have seen, and I have that memory. And then when something new comes, my body is primed or like partially activated so that I can actually respond faster or efficiently. Some people, it's completely new, and then they need to start from from point zero, and then they have to get activated. So everybody's body reacts very differently, and the reason why we are able to block the coronavirus is fully based on the orchestrated interplay between our innate immune responses and the way we elicit our adaptive responses. So it's very tough to kind of give a simple answer, because the truth is it's much more complex. The T cells making antibodies and the T cells organizing this. But I have to caution you something here. And the main reason I'm cautioning you this is because there might be some people in the audience who might have a huge faith in many other alternative medicines, which is absolutely fine. So I have to put a disclaimer that I'm not here to suggest to you that it's a person's complete freedom to choose what kind of medicines or something you need to take. But now, at least in Kerala or in many parts of India, now they are selling immune boosters as medicines, and they do not protect you from SARS-CoV-2. There is no evidence at all. So basically, the Ministry of Ayush has released a report telling that we have now immune boosters, and people have been volunteering to give it to people. So if you don't wash your hands, if you don't wear the mask, and if you just keep taking immune boosters, no, they're not gonna. first of all, the concept of immune boosting is a wrong concept, because you boost it too much, you get actually allergies, and you have hyperactivity. And if you boost it a little lower, you have the cancers. So the boosting, if it's too much, you can even have autoimmunity. Like, that's a very confusing word. So keep in mind that, like, anything you take as immune booster, there is zero science-based like studies which shows that it'll affect in any way. So if I hurt anybody's personal feelings, I apologize here, but that's not going to help you at all. Okay, so coming more on how exactly we handle the coronavirus. So I have been speaking with Dr. Abdul Salam over the phone last time, and we were discussing about how every country handled this. And there are some good examples which we can look on, including our Kerala model itself. At the moment, my aim is not really to say that these countries did good and hence it should be the best case scenario. It's not like that, because not every country has the same working culture or economical scenarios that they can implement this. But some good examples are shown here. So first is Taiwan. So Taiwan's location is very close to China, and in fact, it is partially controlled by China. So when the outbreak happened in Wuhan, Taiwan actually immediately banned all the flights from Wuhan. And they actually did a very early intervention. They made a compulsory use of masks. And surprisingly, they did not shut down their entire economy, but they had the social distancing and extra hygiene and everything. So everybody was wearing masks, everybody was washing their hands, and they didn't go for a total shutdown like how India is doing now or how Canada has done now. And you know what? They actually controlled it very well. Considering how close it is to China and considering how much risk they had, they did a good job. It's a good example to look at. South Korea. South Korea was not only having early interventions, they even had drive-through testing clinics. You be in the car, you just go drive-through and they take a swab out of you. And they were doing massive testing. In fact, South Korea and USA had the very same confirmed positive case on the very same day. And see the measures South Korea took and how things changed, and see what USA did and how things happen now. So it's all about acting very early. Germany, also a good example. One good thing about Germany is that they did a lot of aggressive testing. Like, they did so much of testing. Like, their data actually suggested all the people who was doing modeling that, the mortality rate is very low, because there are so many people who had the coronavirus, but the number of people who died was very low. Even there was an article in New York Times saying that what is a puzzle behind the very low mortality rate in Germany? And there's no puzzle. Basically, we have very low mortality rate. It's all about testing. So you test a lot of people, you know how many people have been infected, and very few people actually die. So your percentage mortality is very low. So now, they also had quarantines, and they have an excellent healthcare system. So they did a good job. USA did not do a good job. They actually largely downplayed the pandemic in the very early stages. There was no early measures. Their kits were faulty. They tried to develop their own kit, which was not according to the CDC or WHO recommendation. They were trying to do that. Basically, they designed faulty primers. Yeah. So if you think about polymerase chain reaction and doing the PCR test, they had faulty primers in the first place, which is like, when you think from a science point of view, you feel like, what? That's what they did. So this is what happened. And they not everybody had access to good testing or good healthcare, massive outbreak. So, and there's also a massive outbreak in New York. New York is like an epicenter because of the number of people coming to New York from all over the world. Like, it's one of the busiest airports in the world, and the city itself is so vibrant and very diverse. So they had every chance of getting it. So the outbreak in New York is something which you cannot expect in any other countries, because New York is New York. No other city is like that. So you have so many people coming there. So they were unfortunate enough to have cities like New York, where all people were coming and many people had coronaviruses, and they were asymptomatic carriers, all these problems. On top of that, they largely ignored all the intelligence advice. They largely ignored all the doctors' advice, and they actually downplayed the pandemic. And that is mainly from the political side, and you kind of get that. In terms of lockdown, India is doing the best job. We have the largest amount of people which has been in lockdown. And even at the time of ongoing outbreak. And China, who has a population above us, didn't do that at the time. It's only at the height of the outbreak they did something like that. So if you think about all the common factor for success, I would put this as an efficiency and the rapid action at the early stages. Massive testing, rapid action at the early stages, quarantine measures, use of mask and everything. So when I told about that aerosol data, like not data, but when I showed that all the slides about how it can spread by aerosols, we didn't know that until I think late March or mid-March. We didn't know the aerosol transfer. The first experiment that was again I forgot which paper they published it. It's only till that time we didn't know about that. So we were telling about droplets and 2 feet social distancing, whatever. But this is all important. Some people just took a precaution of wearing the mask, and that really helped. I saw this nice cartoon from, I don't know who is a cartoonist actually. I think it's Satish Acharya. But this is basically our health minister in Kerala. And you can see that that's a coronavirus like a student. And you can see the scale on her hand. Just saw the scale of efficiency. So we had to really thank our health department because we did pretty much everything. We had experience from the Nipah virus. So prior experience is very important. Our healthcare professionals were actually more trying to kind of prevent rather than cure, because we had limited resources to really actually cure things. So we were trying more on prevention methods. So we did massive, contact tracing. You know more than me, like how amazing Kerala did, and it's something which we can be proud of, and it's really good. That said, again, we cannot think that, okay, the Kerala model is going to work in any other country or any other state, because they have their own limitations. So what is happening in the SARS-CoV-2 India? I'm showing this because it's one of the most visited site, the Worldometer for the coronavirus cases. India is now number 11. So I remember that I was in Canada, and I look in Canada, I always look at the Canada. Canada was at 15 all the time, 15, 14, 15, 14. And India was like way below, like back in March or back in April. And now it's crossed Canada, it's actually crossed China. So you have a lot of cases in India now, which is actually a bit concerning because first of all, we have a lot of people which are living together and they are in very close proximity. So the concept of social distancing sometimes don't literally work in our families or in our communities. It's very tough because we are so many we are a dense populated country, and it's very tough. And at the same time, it is only now we have been doing massive testing, and the more testing you do, you find more cases, and then you can call it as a positive case. And you can, so the testing increases, you're finding more and more people who are actually having the virus. And it's not about something about a pessimistic end, but it is a bit worrisome. Like if we don't take it very seriously within our community, it can be very challenging, because based on the modeling we have again, modeling is never true fully, I will come to that. Based on the modeling we have, it could be that if you take a trajectory of the US or UK, we could be going in a very bad way. All models are wrong, but some are useful, by George Peacock. He's a very famous British mathematician. And the reason they're all wrong is because we depend on something called the R0 of the virus. So I have a video which I'm going to quickly skim through. So basically the R0 of the virus is nothing but the basic reproductive number. So how many number of people can you infect? Which is very hypothetical if you think about it. You cannot really measure this. Like there's no like an instrument to measure. Like, you just go and mingle with people. Between the time you and you have to assume that everybody is susceptible. That's an important thing. When you have a new pandemic, a new virus, you have to assume everybody is susceptible. So this is what happens. And you have more and more people getting infected. And they have been doing so many modeling on this. I just want to quickly show the video. So you can see that when you have the low R0 values, that you're only infecting very few people, you can see the spread here. And if you just make the relaxation, like, if you're just not maintaining social distancing, or if your virus is actually having double the infection radius, just see how fast things are going to spread. You see all the dots are basically the kind of, people moving around, and you can see how fastly you can spread the whole community. So it's like, just increasing the area twice, or just increasing the radius twice, you are actually going to infect a lot of people. So the so-called R0 is even though epidemiologists use it a lot, all we can learn from this is that if the R0 is greater than 1, it's an epidemic. And if its R0 is 1, it's basically endemic. And anything below is means that you're actually controlling it. So that is pretty much we can learn from this. In the beginning, we have now data 2. The latest data suggests that we are much above 2, because we have a lot of people with antibodies who never had symptoms, which means they had a coronavirus, which means these have been transmitted. So our modeling has always been incomplete, mainly because of asymptomatic transmission. That is the biggest challenge in that. and the lack of testing. So only if you have enough testing, and if you were to catch in the very early phase, you can do any modeling. And since there is so much challenges in modeling, we cannot really say, whether the social distancing is going to really help. The long story short is the 5 points in that thing. You have to have social distancing, you have to have good hygiene, you have to limit the traveling, like traveling across countries unless it's you have to go for quarantining. The minute you have a case, isolate and quarantine. All of them has to work together. Only then you can reduce the cases. The biggest problem now is basically the panic. So this is a good cut I found. The real danger is only very less, but we have been creating fear and panic and hype, misinformation, overreaction, hysteria. But thanks to our news channels or, like the politicians behind this. And so this is another important thing which prevents all this. So what is our real future? So everything I told was about the coronavirus and how we are actually heading towards. So we have two fates now. We still going to spend few years wearing masks until we have a vaccine, unless we have a natural herd immunity, or the virus surprisingly disappears from the community, which is very, very unlikely. Or we can see some sunrise, like if we have some hope, we have a vaccine and we can move forward without masks and so forth. Everything depends upon a second wave. So what I'm showing you is basically the second wave from the Spanish flu. The fun fact, Spanish flu didn't start in Spain. Okay, so that is again interesting. That like Spain was the only country which was reporting properly about the virus, and hence they nicknamed it as Spanish flu. But we still don't know the real origin of the Spanish flu. Anyway, what you can see here is the first peak. We are actually here with coronavirus. So for the Spanish flu, what happened is that we had a second wave. You can see that it killed more people, and then you had a third wave, and then only it subsided. So we cannot completely lift our restrictions and rejoice. This is what is happening mainly in North America and in Europe. Like people are slowly removing restrictions, but they're also equally worried about a second wave. So our real future depends upon whether we are going to have a second wave or not. And can anybody tell it accurately? Yes, Dr. Fauci. And according to Dr. Fauci, unless the virus is completely removed from the community, there is still a chance to have a second wave. So how do you remove the virus from a community? Either having a vaccination or having a herd immunity. So there is a good chance we might get a second wave. And you already told that some of us are already seeing a second wave in India itself. The problems we have now is increased anxiety and stress. People have died of COVID. People have lost jobs. They have lost businesses. They have delayed graduation. You're giving all exams in a different way. Like, now here in my own university, people are doing masters and bachelors. They've been thinking, should we give a full master's degree because he or she didn't do the practical courses? So shall we give him like a partial master's, because you have to see a YouTube video how to do a Petri plate or how to grow cells in culture and so forth. So these are all challenging times for both students and also for the teachers and also for the parents. But this is what happened. Broken relationships. We are in a time of very high anxiety and stress. And another important thing about it is that after the COVID, we have a research explosion. So there are so many COVID-19 related work coming every day. The scientists are unable to catch up with the new developments happening. Like, it's impossible. So there are so many preprints in bioRxiv. bioRxiv is like a common server where you can put your preprints, which is not peer reviewed or published. Usually when you put in the bioRxiv, you actually also submit to the journals of interest. How do reviewers spend all the time reading this, like thousands of COVID papers coming? It's very tough. I'm almost finishing. And the last thing is about our future, the pandemic pipeline. So we need a vaccine or a herd immunity to prevent, or once we get it, we need an antivirus drugs. We need a convalescent serum or a monoclonal antibodies. So even in our, in the province which I am here, we are having monoclonal antibodies, more than 500 monoclonal antibodies from people who have recovered. So in case in future in India, if you are taking a monoclonal antibody therapy, keep in mind, it most likely would have come from Canada, because this has been a pioneer company, has a unique technology to screen millions of cells, and they have found that. So we have a lot of other strategies to do that. And when you come to vaccines, experimental therapies, something to keep in mind is Oxford vaccine. And many of you might know this, like, the Indian Serum Institute is already making millions of Oxford vaccine, even without knowing whether it works. And actually, the thing is that we now have the data showing that it actually worked very nicely, at least in macaques, and we are already in the clinical trial. And you can check the Serum Institute webpage, and they have all the information. So this is okay. I'm a bit over time, like almost an hour. So I'm just going to wrap up with the other two more slides. So my last thing is about the economy, and I'm not an economist, but what I can say is that like we are going to a phase of secular stagnation. So we have a low productivity, low private investment, and near deflation. So we're going to have a tough time ahead. With also the gap between the rich countries who are having the emerging markets and the rest of the world who suffers is going to be much more widened. And the more time you keep under the lockdown, we're also having a slower recovery because well, think about your economy as a bicycle. Like unless you're not driving it, unless it's not moving, it's tough. And unfortunately, many low wage, low skill in-person service jobs are going to be lost, and it's not going to come back at all. But it's a tough time. It's not a very optimistic news, I can tell you, but this is unfortunately the case. And there's a lot of thoughts on this. Like I put some couple of things on that. We will have a less office dependent culture. We might have more digital, like what we're doing now. Like we talk to each other through Zoom or other apps. You only travel for essential needs. Tourism industry is going to decline. You might have an immunity passport in case immunity is being established. We have a decrease in political polarization. We don't care when you have a common problem. We don't care whether we have a party affiliation, so forth. Increased trust in science and evidence-based medicines. Less individualism, more than thinking in myself. You think about the community, about our family and so forth. Religious worships will look very different from now, and you have to save a lot of money for the future. With this, I want to thank you. And last but not the least, in case if you happen to meet anybody who is in a healthcare professional or in a primary care worker, please explain your gratitude to them, because these are the frontline workers who are helping a lot to keep everything in control. Okay, thank you. And the thing is that like, I don't know what the question is, but we have the aerosolic transmission of the virus. And this is the same reason many people get sick, even though they don't directly come in contact with the sick person. So this has been already been shown for other viruses, and now we have evidence for the new coronavirus as well. So I don't know what was the question. Can somebody point out to that question? Sorry, just catching up with all the chats. Cytokine storm? Yeah. So there.
One of the students asked questions on role of pH.
Yeah, I'm hearing that. Rahila Yakub, right?
Yeah.
Yeah, the question is like, I'll read for everybody. The question is like, you have mentioned aerosolic transmission. So is it an airborne disease? And the answer is, any respiratory viral disease is actually airborne. So when I say airborne, the degree of airborne depends on that. For example, you might hear about people who had chickenpox, and your neighbor had a chickenpox, and sometimes you might get a chickenpox, right? I'm not telling that kind of scenarios, but if there is, so every time you cough or sneeze or talk very loud, you are constantly having a very small droplet coming out of your mouth. And if you're infected with high amount of virus, then there's a chance that this can be in the room. So it depends. If you have a fan in the room, maybe it's just like rotating and just goes away. Maybe if you're in a room without any air circulation or something like that, it can stay. That's why I told the example of an elevator or the lift. You go to the lift and you sneeze inside the lift, and there was no proper sneezing. There was no mask. You just sneezed very loud, and they have aerosols all around the lift. And you just exit to the lift, and someone else come back after like few minutes or few hours even. Theoretically, you can get infected, and that is the case. Like that is why all the countries which have really controlled were using masks, not to prevent it, but rather when you are able to sneeze, you are able to contain it a little more better. So, well, I use the word airborne. I had to be a little more careful here. So, yes and no. So aerosols will be formed only in hospitals. No, that's not true. That is a very common misconception. Aerosols are not formed only during hospital procedures. Actually, aerosols are formed just also by coughing. People have debunked this long time ago with the flu virus itself. So there have been very well controlled experiments with flu viruses, and they have shown that the aerosolic transmission is also one of the important factors of flu virus spread. So our belief that droplets are formed only when we cough and aerosols are formed only for those, that is not actually right. I have heard this in some WhatsApp too. Otherwise, we see big particles settles down easily, you won't travel much disease. Are true. Yeah, I think I answered it. Like, you basically have both. Aerosol is there when you do that, when you talk, when you cough, and so forth. So that is why. So in many places in North America, you can only go for shopping, especially in Costco and some other places, only if you wear a mask. Because if you are infected and if you sneeze, you don't want other people to get the disease. So even though you keep social distancing, we are like staying very far apart. Still, you just wanted to have that just to reduce a chance, because we don't know whether you are a super carrier or not. So it's heard there are 7 serotypes of coronavirus. Cocktail vaccine essential. What is the factors to notice? These 7 serotypes of coronavirus. So when you say about serotypes, I'm not understanding. Are you referring to the strains? Because yeah, I mean, like we have 7 different human coronaviruses, and there was a one point of time where people were doing this so-called nextgene.org, where they look on all the different mutations in the coronaviruses, and they said there are different strains of coronaviruses. I'm not sure what exactly is a question, but assuming that the question is, first to clarify, we have 7 human coronaviruses, which I showed in the beginning, and we are talking about the SARS-CoV-2. And the SARS-CoV-2, even though we have many mutations, we cannot call that it has completely developed into a strain. So you need to accumulate enough mutations in order to call itself a strain. If you have one amino acid substitution, that's fine. It's a long genome, 30 kb, or like a couple of amino acid changes, it's not a problem. It's all about having the amino acid changes in the key residues, and that becomes beneficial also for the virus. Then we can call it as new strain, or else we cannot call it as no strain. And I hope that the cocktail vaccine, see, the thing about vaccine is like, I really went very fast with the Oxford vaccine, but I'll tell you what the Oxford vaccine is. The Oxford vaccine is an adenovirus backbone-based vaccine. So adenovirus, it's a very common virus. We get it's very mild, you get a common cold or things like that. It's a very small virus also. And instead of having the human adenovirus, we are using the chimpanzee adenovirus. Why? Because our body already have human adenovirus antibodies. So you cannot make an adenovirus which is human serotyped or human tropism to raise immune responses. So use a chimpanzee, our cousin's adenovirus, and then you completely inactivate the virus. You make it replication incompetent, and then you codon optimize your spike protein so that we can produce a lot of them, and then immunize. So a single shot of that Oxford vaccine, I went through it very quickly. A single shot of the Oxford vaccine was sufficient to give a protection. None of the monkeys developed pneumonia. They had no viruses in the lungs. They had some virus in the nose, but no virus in the lungs. So yeah, so the so-called cocktail vaccine, I don't know whether we should be looking on a cocktail vaccine or whether you should look for something more specific on, how well you can elicit your immune response. So rather than having many, so the cocktail vaccines are good sometimes for flu, for instance, because the flu, there's so many strains. Sometimes we use cocktail vaccines because we have different, H1, N1, H2, N3, all different combinations. So you use a cocktail. For coronavirus, they don't mutate very rapidly. So the idea is to have the spike protein, or that's the best antigen to be presented, and then to make sure they elicit a good response. And the response also is very different. You have something called Th1 response and Th2 response. The Th1 response is what we need by producing interferons, and Th2 is more like dampening the responses. So you really need to balance what kind, and everything what we have from Oxford vaccine is so promising. And who knows, maybe once the Oxford vaccine clinical trial ends, we don't have to wait for the vaccine to be produced, because the Serum Institute in India is already producing millions of it. So the funny thing is, like how China is called as the factory of the world, India is called as the pharmacy of the world. So we are, thanks to a culture of eating so many medicines, or not eating, but consuming so many medicines, we have been very good at manufacturing in large quantities for a cheap price. So they have been investing on the Oxford vaccine and trying to get as much money as much copies of it as possible, even before testing is done. Okay, there's another good participants raise your hands. Please send. Okay, but the airborne transmission. You mentioned airborne disease. Yeah, how much distance? I think I answered that.
One of the Libin.
Yeah, can hear you.
one of the student, Jagan, asked about loss of biodiversity, maybe the issue for zoonotic diseases.
Loss of once again?
Biodiversity. Loss of biodiversity, maybe the cause of zoonotic diseases.
Yes, absolutely. Absolutely. No question about that. So, like, as I told you, bats are not the reasons why we get the disease. We are the reason why we got the disease. So we have been interfering into a bat ecosystem. And obviously the bats, as I mentioned you, is like super host of all these viruses. And in China, there's also this tradition of eating bats as well. So they have been used in the trade market for eating. And and we have been deforestation and agriculture, our agriculture practice of deforestation and encroaching into the bat ecosystem is definitely the main reason why we are getting more. As I told you from the little graphical representation, we have been together with bats for a very long time. We didn't have any bat viruses coming spilling over to humans. Only then we went to their territory, we got it. So there was a question actually about immunoboosters. I want to ask, how can you, this from the Divya case, how can you say immunoboosters are helpless? So this is something which I want to tell you. First of all, you have to define immunoboosters. As I told you, like, that terminology itself is very finicky. Like, I'm an immunologist, and when you say that immunoboosters, what are you even trying to say? Are you going to make a B cell make more antibodies? Are you making a T cell make a severe cytokine response? So that word itself is very confusing. If you're telling about having a healthy lifestyle, having a good diet, doing proper exercises, washing your hands properly, and like everything else which can naturally improve your immunity, I am with it. That is a way to boost your immunity. You can you can actually slowly reduce the consumption of fatty foods from your diet and everything like that. But you cannot give a pill and say that that's going to boost your immunity, because there is no scientific evidence for that. And the Ayush ministry, which released a paper, and it's very funny because my cousin is a homeopath in Dubai, and he is the one who sent me the article. It says that when you use arsenicum album, the paper was completely different. The paper was about using arsenic and showing that there is enough kappa B activation, which is actually like causing some inflammatory, it's like immune response. But the dosage they used, and this is basically studying arsenic contamination in water. So it destroyed the thymus, so which is very bad. You have thymus destruction happening, and the paper was completely irrelevant to the Ayush ministry citation, and I was like shocked. Like, how on earth can you advise millions of people to take a pill and say that it's going to boost your immunity? No, there's no magic. Like, I would be happy to know the science behind it. Like, you can say immune boosters are helpful, but you have to provide me some scientific validation to that. There is zero scientific validation to say that there is an immune boosting capability. That's a truth. That's very tough to accept in the beginning, but that's a truth. How can work plasma techniques? So there's a question from Sunil Sikawar which says, how can work plasma techniques? So I'm not able to fully understand that question, but if the question is regarding convalescent plasma as a therapeutic method, the idea is that I got COVID-19 and I recovered completely. So my serum is full of antibodies. So basically I can donate my blood and people will isolate the serum, and then they will check for all the infections, like whether I have HIV or hepatitis or anything. And then, just like your blood matching, or when you donate your blood, and when there's another person who is actually having similar disease like COVID, then when they are in the critical stages, when their immune system is not able to fight enough, and when the virus load is going up, then my serum can be helpful. So I hope that is a way you're asking, how can work plasma techniques. So our plasma contains antibodies. The antibodies are proteins which bind to the virus particles and prevent viruses from doing all the harm. So this is how we have to think about it. One of the best webinar which thank you.
Libin, one more. Hello.
Sure. Yeah, go ahead.
One Dr. Mercy teacher, he asked that whether there is a chance of increase in coronavirus during rainy season in Kerala.
Yeah, that's so my honest answer is I don't know. The main thing is that, like, so here's what we think. So in North America, every time you have a flu coming, usually towards the beginning of winter. So when you already have a flu, on top of the flu, if you have corona, your mortality can slowly go up. The severity can only go because your body is fighting to flu, and then you have on top of that corona, which is going to be very bad, right? So if you think about similar thing in, whenever we have the rainy season in Kerala, we sometimes fall sick, right? Due to many other viruses and so forth. So our immune system is slightly compromised. And on top of that, if you get a coronavirus, the way our body handles the coronavirus when we were completely healthy versus how it handles when we're already fighting an existing rainy season virus, it's very different. So there could be a chance that in Kerala, when there's a rainy season coming, and we have seen that we get different kinds of fever, some people call Taliparamba or other things, we don't know. So if we get it, then it's going to be very bad. We don't know. I cannot say with 100% certainty, but there's a good chance that it could be there. There's another question, like, while antibodies IgG and IgM against novel coronaviruses generally develop in 1 to 3 weeks infection, some people seem to develop humoral response as little bit to antibodies. What this happens? Let me just repeat that question. It's from Anandu SP. While antibodies IgG and IgM against novel coronavirus generally develop in 1 to 3 weeks infection, I'm assuming it's at 1 to 2 weeks infection, some people seem to develop a humoral immune response as reflected by detected antibodies. Why that happens? So here's the thing. So when our body makes antibodies, right, the idea is that, like, you have this protein which binds to a specific structure on the coronavirus, right? So usually this is very specific. Even though I say specificity, there is also something called cross-reactivity too. Okay, that is something about antibodies. So that is one of the main reasons antibody testing cannot be fully believed, for the fact that you have cross-reactivity. So as I told that, maybe there are some people who had a humoral immune response developed to some other closely similar three-dimensional structure. Okay, and hence they can actually have these antibodies in circulation. So some people seem to develop a humoral immune response developed by digital antibodies. So digital antibodies, okay, it's good to have the antibodies there. Sorry, I'm not going to fully get the question. While antibodies generally develop to them, some people seem to develop immune response to digital antibodies. Why this happens? Is Anandu SP there? Can you just repeat that question one more time? Just type that question one more time. I'll just go to another question if there is there. Thank you so much. How are the asymptomatic carriers occur in population? So tomorrow there's this second part of this talk by Anurag. So Anurag will be describing a paper which is published in Cell, and they have described the mechanism of why some people have asymptomatic transmission. But in order to quickly answer this question, we can think this from the part of the immune response itself. So my body might be taking this. I might be infected with a very low dose in the first place, and I have a very strong immune system. So I'll be just like, fighting the virus. The virus will be there in my nasal swabs and maybe in upper respiratory tract. But when it comes to the lungs, it's all taken care and cleared out. Even in the vaccination trials, the monkeys were fully protected from pneumonia, but the monkeys had viruses on the nasal swabs, on the nasal areas. So it suggests that, like, even though you are immunized and you can be still getting a coronavirus, you can be still transmitting it. Strange, right? Yeah, that's okay. So the answer to that is like, where's the question again? Oh, where is it? That is the reason why asymptomatic transmission is mainly. I think I lost that question. Yeah, and do they spread disease? Absolutely, yes. So when you say asymptomatic transmitters, I am an asymptomatic. So this is what happens. Like many people lost their parents because they were actually positive for coronavirus. And they went and met their parents, and their parents were, first of all, old. They had a weak immune system, and they have also 1 or 2 comorbidities. They got it much more severely, and they couldn't make it, and they died. So this is one of the main reason I'm emphasizing that, like, even though you might seem totally perfect, you are absolutely healthy, you might be a carrier, and that is a very bad thing you're doing to the community. As a responsible citizen, it's very bad. There was a question about Ayurveda, which I would actually like to read.
So, Libin?
Yeah.
One question from one student from Myanmar. Can blood serum really respond to this virus?
Can blood serum
really respond to this virus? Student
Can
from
you just
Myanmar.
Can you please repeat it one more time? Can blood
Can
serum?
blood serum really respond to this virus?
Well, when you say respond to the virus, the idea is that like so the blood serum contains antibodies, and antibodies gives only passive protection. Okay, so think about the mother and the newborn baby. Okay, the newborn baby doesn't have the full protection, and the protection is coming by mother's immunoglobulin or antibodies, which is coming through the breast milk, right? So there is a lot of passive protection for the newborn baby, purely coming from the mother through her acquired immunity and the antibodies in her body coming to the breast milk. So you can think this as a similar scenario. When you think about antibody-based therapies, it's all passive protection because it is your body is not really fighting. There are some soldiers made by someone else. You're just using their soldiers. That's a way to think about it. Yeah, and convalescent plasma has been shown to be one of the very effective methods to treat some people who are actually advancing. Why the mortality rate in India lower to European nations? Is it because of viral strain? So thanks. That's actually a question from Vigneshu. So this was a thing which I have discussed it. So you can only know the mortality rate only if we actually do enough testing. So when people were looking on mortality rate of the global mortality rate and the mortality rate only in, for example, like country like Germany, suddenly found a surprise. Like the Germany was doing massive testing and few people died. So I'm not sure if India's mortality rate is very low. I simply don't know, but it could be that we might be getting probably, as I said, maybe a less virulent strain. Again, I don't use the word as strain because the virologist actually has been strongly advising against using the word strain. Even though we have enough mutations, we cannot call it a different strain. We have so many mutations in the virus. It's different from the Wuhan virus and what you have in Europe or everything. Virus has already been mutating even within a body itself, but we cannot call it a virus strain. It could be different, a weakened virus, or it could be because of many other reasons, like maybe it's tough to say why exactly. One possibility could be the testing. We really don't know. And one more important thing is about how you report death. So think that you're a person who has heart problem, and then you got COVID, and then you died. So in the post-mortem report, the rule in Canada is that you're right, person died of heart attack, but the person was also having COVID, and you will count it as a COVID death. Many countries has different policies. So some people just die in the homes, and they're called natural death at home. We don't know. So it all depends. So it's very my general principle is that it is not good to compare the mortalities happening in different countries, because it's very different. Every country has a different metrics or standard or threshold of doing things. They have their own protocols of what is called a natural death, what is called a coronavirus death, and everything. So that is very challenging to do that. On the contrary, what is good to compare between countries is which countries were actually able to control it properly and what measures they took. And I was discussing the same thing with Dr. Abdul Salam before, that in Corona we had this. Sorry, in Kerala we had the Corona booths. And how did we had the Corona booths? It's actually initially from South Korea. South Korea had this wonderful idea of having Corona booths, and then Kerala immediately took it up, and that's how we actually got it. So it's good. So I think my answer to that is like I still don't know why it's a low mortality rate. Whether India might be having a similar mortality rate, we don't know. How the mortality is being assessed, what is the criteria for calling COVID death versus a heart attack or something else, a natural death at home. And second, enough testing. Are we doing enough testing to know whether it's the case? And different strain, I would not call strain. I would still say that maybe there might be a we don't know whether our immune system has been. We call this term called primed. So it's a very common term used by immunologists to describe something as partially activated. It's not like a fully resting state. It's not a fully activated state. It's something called partially activated, called primed. So in our body, the more primed cells are, which you can also get by vaccination, also things, or you can also guess by pure environment you are. Like if you live in a little more, the hygiene hypothesis, right? You need to have an environment which is actually you're constantly your body is triggered. You're constantly producing antibody. Your body is constantly doing that. You might be able to probably clear some things away. On the contrary, if you live in an extremely clean, dust-proof, fur-proof place, people develop allergies, and it's shown that it's not very good. So it could be all of these reasons. Between the how much lockdown is useful, then how can we develop immunity? It's okay. So this is a question actually from Supriya Shikalgar. I cannot say how lockdown is useful, but I can say that if we did not have lockdown, we would have way much higher deaths. No question about that. So India was going in an extremely bad trajectory, and the lockdown actually helped in reducing it. But it was not extremely successful because the lockdown was not like, see, for example, the lockdown in China was like on a different level. The government there is very like the Chinese government is very authoritarian, and they really make sure that nobody gets out of the house. They use drones, they use camera surveillances and everything, and they go for inhuman measures to really make sure that people don't leave the homes. They look on your cell phone data and everything like that. We are not doing anything that crazy. Like, we are still doing within certain limits and boundaries. And so I still think that it was useful. Then how can we develop immunity? Like, the immunity can only be developed either by getting the virus and you recover from it, which is a chance, because what if you get a very strong version of it and then your body was not ready to fight for it? You will not recover. Rather, sometimes you go to severe cases and you might lose your life. And the second option is vaccination, which we had actually waited. So there's a concept called herd immunity. And, the Sweden was actually trying something called herd immunity, and UK tried in the beginning, and failed. And that's why you have so many deaths in UK now, because UK actually, the herd immunity, some of you might know, is that like, as if there is a sufficient number of people in a community which are actually immune, and the virus doesn't spread. You see all this modeling I showed, the people moving around. You cannot spread to another person because that person is like immune or resistant to the virus. So yeah, you can only develop immunity by either by vaccination or by natural exposure. But going the risk of natural exposure to get an immunity is probably not a wise idea. There's a question from Tamanshal Monda. Between the PCR technique and the antibody technique, which test is taken less time to get the result? Well, I think the lesser time is obviously the antibody testing test, because the PCR technique needs first the conversion of an RNA to the DNA, and the DNA has been amplified, so it has to go through enough cycles. But we have been actually not having like 30 cycles of PCR. We can actually do like, say, 10 cycles of PCR and see the initial, the gap. So the negative control will go like this, and the positive control will go like that. So if you go wide enough, you can see this nice difference. But in the very beginning itself, you can sometimes in 10 cycles, you can see the amplification. So depending upon how many cycles of PCR, but in general, I think that it's antibody test which is faster.
So, Libin, Dr. Libin?
Yeah.
I think you have answered many questions. So all questions from the chat box you can answer. So
that's true. I mean, like,
I think
oh my God. I didn't see all these questions coming. Sorry. Yeah, I feel now we're bad. Anyway. Yeah, I mean, maybe the moderator can look into this and maybe pool the questions, because some of the questions might be overlapping with each other. I saw somebody asking about Ayurveda. I just was clicked on that. So that is what. Again, I'm not to advise that you should not do this, but there is no science-based proof for that. Immune boosting or any supplements is going to help you from fighting better. Absolutely no. Have a healthy life, have a healthy diet, do the exercise, do the things which actually makes your body healthy, but not by taking. If you have any micronutrient deficiencies, like an iron deficiency or like selenium deficiency, yes, that's a different thing. But if you don't have any major deficiencies, then there's no point of doing that. The malaria transmitted mosquitoes.
So I think that okay. We are now in the end of the session. So if you have participants, if you have too many questions, you can raise tomorrow. Tomorrow also we have a similar kind of presentation.
So many questions.
So with that, I would like to call Bushra, Assistant Professor, Department of Zoology, Syed College, for vote of thanks.
Okay. And thank you, sir. A very good morning to all. Honorable Principal of Society College, Dr. Ashraf Kulli, sir. Respected Chief Guest of the Day, Dr. Abhilash, the District Nodal Officer of COVID-19, and Dr. Libin Abraham, the Senior Scientist, British Columbia University, our IQAC Coordinator, Dr. Nafisa Baby, and other members of the IQAC, Department HOD, Dr. Mundas TMV, and dear colleagues and dear participants. First of all, it is my privilege to propose a vote of thanks in this occasion. Thanks for that. So on behalf of Syed College, Department of Zoology and entire team, I wish to express our sincere gratitude to Dr. Abhilash. Thank you so much for joining us. You are a real hero in this war, and thank you so much, sir, for sharing your time. So next, I would like to express our sincere Libin, sir, for joining with us, for sharing your knowledge and experience with us. Personally, it was a wonderful session. We know tremendous fake news is related to this COVID coming day by day. So hearing from a real or a genuine resource like you is very important in this situation. So thank you so much for joining us. And further, I must mention our deep sense of appreciation to all the participants from all over the world. Thank you so much for your kind cooperation with us, and hope you all are enjoyed the session. And finally, I also wish to express our gratitude to our institution management, our beloved principal, Dr. Ashraf Kulli, and our IQAC coordinator, department faculties, and the entire staff of this institution for their support and encouragement. Thank you.
First and foremost, again, thanks again for the organizers for giving a chance to talk here. I shared my thoughts on this, but again, some of you might have a different opinion about, probably the origin of coronavirus, or probably the use of immune boosters or things like that. Feel free to, everybody has a freedom to think what they want. But at the end of the day, we're all having the same challenges, and I think we should all try to look on more science-based, evidence-based approaches to fight it, rather than beliefs or anecdotal evidences, like we call in Malayalam as Anubhava Sakshingal, like 1 or 2% says it works. We should not go with that. And first, and I don't know how many participants were there today, but I think I made a record by giving the talk to the largest number of participants, because I gave a class here in Canada, and the strength of the class was close to 300 students. So I think if there are more than 300 today, then you guys helped me beat my personal record of talking to the largest number of audience. So a big thanks to all of you for listening carefully. I was a bit fast in the way I spoke, and if anything was not clear, I apologize for that. But I'm happy to share the slides or information if you need. And if you have any questions which I was not able to answer because of the massive amount of questions I had, if moderator can pull these questions, put them together, and then I am happy to answer these questions. Thank you so much again, organizers, all the teachers, all the students. This is great. This was fun.
Okay, thank you. Bye all of you.
Yeah, bye.